In this lesson, we're going to talk about blood grouping and blood types. So I'm sure that you've all heard of all the different types of blood groups. So you've got like group A, B, group AB, and group O. And so we're going to get into more of the specifics about why this is the way it is. So what we look at is something called a specific glycoprotein found on the surface of the red blood cells, and it gives the red blood cells its ID. So more specifically, group A has a specific type of glycoprotein on its surface, and group B has one, group AB has one, and then group O has none, and we'll get into that in just a second. But this specific glycoprotein is called agglutinogen, and what it is it's an antigen. And when we talk about groups, we talk about the A antigen being on group A. So group A blood types have the group A antigen on the surface of the red blood cell. For group B, it's going to have the B antigen on the group B blood cell. Then for group AB, it has both A and B antigens. And then group O has none. And this plays into this antibody that's in the plasma. So this the relationship between these is an antibody and antigen relationship, and that is really important here in a second. But with this antibody called agglutinin, it acts as a bodyguard. And what happens is, so let's look at group A. So group A has an A antigen attached to the surface of the red blood cell, but it has the anti-B antibody in the plasma. So for instance, if in blood transfusions, where we'll talk about this in just a few minutes, but this is where it comes into play. So group B, if group B was introduced to a group A patient, this anti-B antibody in the plasma would see this antigen show up, and it would then cause a reaction. And this reaction is really important. This reaction is called agglutination, which is a clumping of red blood cells. And the reason this happens is because it's a reaction between the antigen and the antibody of the same ID. So when we talk about antibody A and then antigen A, that's a bad thing. So the body's response, the body sees this, for instance here, in the anti-B antibody for group A, it sees this antigen as a foreign pathogen, and what it wants to do is break it down. So if we move along, we see, okay, look, group B, it actually has an anti-A antigen. So if a group A blood was transfused into that person, they would have this agglutination happen. So for group AB, it's a little bit different. So if they were to get A, cool, they don't have the A antibodies in there to fight off or to perceive that blood type as a foreign invader, so it's not going to have a reaction. But then it's going to be the total opposite for O. They have both. So if either of them show up, and we're going to get into that in just a minute, but if either of them show up, that's where it comes into play. When we talk about blood transfusions, we want to talk about a couple of things. First off is the goal. When we are aiming for a goal for blood transfusions, we want to transfuse blood from the same group to the same group when possible. And if that's unavailable, you want to use groups that don't have the same matching antigens, antibodies. So for instance, group A should get group A, group B should get group B, group AB should get AB, and group O should get O. So let's look at this, for instance. So if we start with group AB, they have both the A and B antigens all the way around. So they have no antibodies to recognize as the foreign invaders. So A can come and transfuse on the A, A can transfuse on the AB, B can transfuse on the AB. Well, if we go the other way, if you notice the arrow doesn't go this way, it doesn't work that way because of this B antigen that's on the surface of this red blood cell. So that would actually cause a reaction here. But if we go, if we look kind of the other way, let's look at if we were going to transfuse O to AB. O doesn't have any antigens, so you're not going to have any of that adverse reaction happening. Same thing for A. So A only has the A antigens on it. It can't receive from AB because of the B. It cannot receive the B because of the B antigen. And then O, it can because there are no antigens on the surface of the red blood cell. So if we even look at O here, O can't O has no antigens, but it has both antibodies. So if A tried to transfuse to O, you would have an agglutination reaction. B, if they tried to transfuse, it would be, you would have a agglutination reaction. So that's why it's important to make sure that you're trying to, that your groups don't have the matching antigen antibodies, because that will result in the reaction. So there's this other really important factor called the Rh factor, which is the D antigen. And on the surface of the red blood cell, you have your AB, you have your A and B antigens, but then sometimes you'll have this D antigen. And what this D antigen is it's also known as the rhesus factor. If the factor is present, so let's say this is a patient, they have A, B, let's say they're AB, and then they've got this D antigen, right? So they've got all of them. So this would be AB, and if the factor is present, so here's D, here's D, here's D, it tells us that it's present. That means that patient is Rh positive. So this patient would be AB positive. So if we went and had another red blood cell, and they had A, B, and then went all the way around, but they didn't have this D, and that factor would be absent. So that means that that person would be Rh negative. So this patient would be Rh negative. Now, it's ideal, similarly to the blood transfusions, if you have an Rh-like blood type, you want to give the same thing. So you want positive to go to positive, you want one negative to go to negative. Now, just like with the other blood types, if there is, it's okay to give negative, an Rh negative blood to an Rh positive patient, because they don't have this antigen, therefore they're not going to have that antibody that's going to react to it. But if you go the other way, you are for sure going to get that reaction. So this can't happen. So it's always, you can always give negative to positive, but you cannot give positive to negative. Okay, so let's recap. We talked about the different types of blood types. You've got A, B, AB, and O, and then the antibody-antigen relationship. The antigens are present on the surface of the red blood cell, and the antibodies are in the plasma. Agglutination occurs because of the clumping of the red blood cells, because of that antibody and antigen relationship, or that reaction that could potentially happen. The Rh factor is that antigen D, and if you see it, if it is present, that means that it is positive. And if it's absent, then it's negative. And then blood transfusions. We always want to try to give the same type and the same Rh factor whenever possible. So that's it for our lesson on blood grouping. Make sure you check out all the resources attached to this lesson. Now go out and be your best selves today. And as always, happy nursing.